All right, so today we're going to talk about, I'm going to talk a little bit about Chapter 3. It's titled The Molecules of Life. Just to reference, we have already discussed the basic chemistry principles that underline the biology concepts that we'll be talking about. And we're going to use those chemistry principles that we talked about in Chapter 2 today to build some macromolecules, so some essential molecules that we all have. And then in the future, we'll talk about how we break down these molecules in order to obtain energy for them to perform everyday activities like talking, breathing, walking, and so on. All right, so my first slide says why even study carbon, right? And so you may be familiar with the term organic, inorganic, or non-organic. In terms of biology, when we talk about the term organic, we mean that it's from some type of living organism and that it's composed of carbons and hydrogens. And you'll see down here, the cells are made up of about 72% water, which is why we spent so much time talking about water in Chapter 2. It's obviously essential. About 3% different types of salts, and 25% of your cells are made up of these carbon compounds. And there are four that are in particular that are important that we'll talk about today. These four macromolecules, so these carbohydrates, lipids, proteins, and eventually nucleic acids. So about 25% of our cells are made up of these carbon-based compounds, so carbon is essential, very important element, and it's very important and essential for these four major macromolecules, which play important roles within our cells and ultimately within our bodies. A little review on what carbon is. So remember, we talked about it. We saw it on the periodic table in Chapter 2. Carbons are a very versatile building block, so they're kind of like tinker toys. We can build long chains and add things to it, add functional groups to it to give it different chemical and physical properties within the body. They have very unique bonding properties. Carbon will form four stable covalent bonds, so the term stable here is very important. If they weren't stable, they'd be broken down all the time within the body. Carbon also has 4 electrons in the outer shell. So here's my carbon atom, and here are the 4 valence electrons. So again, carbon has 6 total electrons, but it has 4 valence electrons. So here's those first 2 electrons in that first energy level. And to review from Chapter 2, remember that first energy level of an atom can only hold a maximum of 2 electrons. And then the remaining 4, here they are, 1, 2, 3, and 4. So we have 4 valence electrons. And remember that all atoms want 8, right? So they want 8 valence electrons in order to be chemically stable. They're either going to steal or gain those electrons by forming ionic bonds, or they can share electrons. And so carbon has 4 valence electrons, but it also has 4 open spaces, right? So what is it going to do? Is it going to gain or lose? Well, it's kind of a wash, right? So because we have an equal number of electrons to gain or lose. So carbon, in terms of the biology that we're going to talk about, will always share electrons, right? It's always going to form covalent bonds. And these covalent bonds are going to be very stable. They're not going to have positive or negative charges, so they're going to be very stable within the body. Carbon can share its electrons with other atoms to form up to 4 covalent bonds, right? So it can form a bond here, here, with this electron, or with this electron. And these are some other models down here that you could see carbon potentially take on. It's the same thing, right? We're just using a different model. For this one, you can see here's my central carbon, and here's my 1, 2, 3, 4. It happens to have 4 hydrogens here, but it can actually form bonds with anything, right? Or different types of elements, and we'll talk about that in a little bit. Carbon can use these bonds to attach to other carbons, which forms an endless diversity of carbon skeletons. So you can see carbons can vary in length right here, right? You can see I have a 2-carbon molecule here, I have a 3-carbon molecule here. We can also have double bonds within the carbon, which can give it different characteristics. The carbon double bonds can also occur in different locations, as you can see. We can also have branches which come out, so this would be considered an isomolecule. And then lastly over here we can also have these carbon molecules form these ring structures. So the point here, guys, is that carbons kind of act as, and particularly these hydrocarbons, act as these tinker toys. They're these building blocks where we can vary in length and we can branch off and we can add different functional groups onto it, or we can form circles or longer chains. We can make these chains really long. We can also break them down very quickly. So this gives us a lot of versatility to start with something, a foundation of a hydrocarbon, and then to add on and build different things onto these carbons in order to create different molecules like carbohydrates or lipids or proteins or nucleic acids. All right, my next slide introduces you to this hydrocarbon. So hydrocarbons are the simplest carbon molecules. They're a combination of hydrogen and carbon. The simplest hydrocarbon molecule that we have is called methane. And you can see over here on the right-hand side, here's the simplest carbon molecule of methane. These all represent the same molecule, right? So here's my central carbon, and I have 4 hydrogens on the outside. This is your structural formula, your ball and stick model, and then eventually your space filling model over here. Okay, question says, are these polar or non-polar? Well, these bonds right here, do you see any positive or negative charges? Nope. And they're sharing, so they're definitely covalent, but they are non-polar. They do not have positive and negative charges. So if they don't have that positive or negative charge, is water going to be able to dissolve them? And the answer is no, which therefore makes them non-soluble in water, right? So remember when we talked about water being a solvent? And I showed you a diagram of the negative oxygens and the positive hydrogens surrounding those different sodium and chlorine atoms. Remember, it was those negative and positive charges which allowed something or an ionic bond to dissolve in water. There's no negative or positive charges here, so water will not be able to dissolve them. Therefore, they're also considered to be hydrophobic. So remember, hydro, water. Phobic means they don't like water. They're very stable, very little attraction between molecules, and it's a gas at room temperature. Again, this is just the methane. Larger hydrocarbons that we may see, and we'll talk about these in the future, could form fuels for engines. We can also see long hydrocarbons here, which make up fats. You guys should know by now, right, that water and oil or fats and water do not mix together. And the reason being is because water's polar, and these hydrocarbons, which I'm showing you right here, are non-polar. So they don't dissolve, right? Which is why these fatty foods would not dissolve in water. The unique property of organic compounds depend on not only its carbon skeleton, but also the atoms that attach to that skeleton. So the atoms that attach to that carbon skeleton are going to determine its unique characteristics and properties. So what does it do? Is it a carbohydrate? Does it act as an acid? The groups of atoms that usually participate in chemical reactions are called functional groups, and 2 common ones that we'll see today are the hydroxyl group, which is represented by the OH, and a carboxyl group, which is represented here by a carbon double bonded to an oxygen. And again, there's many other functional groups, but these are just the 2 that we're going to pay particular attention to when we talk about our macromolecules today. So we've talked about carbon, right, and the basics of a hydrocarbon. Now what do we do when we start putting all of these carbons together? We get something called macromolecules, right? And so a hydrocarbon is just a molecule. Macro means large, right? So you can imagine that a macromolecule is going to be a very large molecule made up of many components. These are smaller molecules which join to form larger molecules, and they link monomers to form polymers. So mono means one, poly means many. So when we link, it's just like a building block, right? If you had tinker blocks, when you link these individual units together, you form these large polymers. Of these polymers, there are 4 categories, which we can also call macromolecules. And again, they're carbohydrates, proteins, nucleic acids, and lipids. We will be talking about each one of these macromolecules. We'll talk about their function within the body. We'll talk about their structure as well. So before we look at each one of those macromolecules, let's just define again what a polymer is, right? So remember I said poly means many. Polymers are long molecules built by linking chains of repeating smaller subunits, right? So the mono is the smaller subunit. So if you look at this diagram down here at the bottom of the slide, you'll notice here's a very short polymer right here. It's made up of 3 individual monomers. Here's an example of an individual unlinked monomer, and this would be what we consider a longer polymer. So we haven't defined what these monomers are yet. The monomer is going to be defined based on what the macromolecule is, right? But the same basic process here of linking these individual monomers to the polymer to form longer polymer chains is a commonality amongst all of those macromolecules. Most macromolecules are polymer. Again, this is a repeat. It says polymers are made of stringing together smaller units called monomers. And there are 2 types of reactions here that we're going to focus on, guys. How do we make these macromolecules and how do we break them down, right? So to make them is going to require some energy. Why would we want to break down a macromolecule? Well, if we eat, let's say, carbohydrates in the form of potatoes, all of those individual chemical bonds that are holding those monomers together contain a certain amount of energy. We need to break that energy break that bond in order to release that energy, so that way your body has energy to perform functions, right? Like exercising and walking and talking and breathing. Those 2 types of reactions are called the dehydration reaction and the hydrolysis reaction. So in the dehydration reaction, this links 2 monomers together and you remove a molecule of water. For a hydrolysis reaction, we break the bonds between the monomers, we add a molecule of water, and this is essentially the reverse of a dehydration reaction. You'll notice I also have these little play buttons right here. They will link you to an additional animation to explain this further in greater detail if you need it. And just to remind you, if you do want to play this animation, you need to be in the slideshow mode of your PowerPoint. You're not going to be able to link to it right here because it's a Captivate file. And you won't be able to link to it in the notes form, but you'll have to put your PowerPoint into slideshow mode and it should take you out to that link. If you have any problems, just ask me in class. All right, so here is a nice diagram of what I just defined for you as a dehydration reaction and a hydrolysis reaction. So let's look over here first on the left-hand side. Here's my short polymer. Here's my individual monomer, right? Remember, because mono is 1. And notice here, here's an OH group. Remember that functional group I told you about a few slides ago? Here's an OH, which is an oxygen and a hydrogen. And then on the monomer, there's an individual hydrogen. Through a dehydration reaction, water is released. Where did this water come from? H2O means there's 2 hydrogens and 1 oxygen. It came from up here. Look up here. Here's your 1, 2 hydrogens, and here's your oxygen. So they combine together to release water, and a bond is formed, right? So this requires energy. We can do this many times, hundreds or thousands of times, to keep building this polymer. And notice, guys, the key here, it says dehydration. So when you dehydrate something, what do? You take water out, right? So like when your cells are dehydrated, you've removed water. Well, that's exactly what you've done here. So by definition, a dehydration reaction is used to build polymers. On the right-hand side over here, here's an example of a hydrolysis reaction. Guys, hydro means water. Lysis, to lyse something, means to break something. So it's the opposite of that dehydration reaction. Here's water. Water actually attacks that bond, and the 2 hydrogens and that 1 oxygen then add itself to the end of the shorter polymer in that monomer, so follow the hydrogens and oxygens, and energy is released, right? So just to review by definition, dehydration reaction requires energy. You build polymers. Over here on the right-hand side, hydrolysis reaction, we break bonds and we release energy. This process of dehydration and hydrolysis is going to apply to the building and breaking down of all of our macromolecules. So whether we're talking about making a carbohydrate or breaking down a protein, doesn't matter. The same chemical reaction occurs no matter what the macromolecule is. Here's a practice question. Go ahead and pause it, take a minute, it's a little wordy, answer the question and we'll talk about it in class. Again, our large biological molecules. We're going to start to talk about these carbs, lipids, proteins, and nucleic acids. So our first macromolecule is a carbohydrate. You'll see it says carbohydrates are sugars, or sugar polymers, so long chains. They include small sugars, which you find in soft drinks, longer starch molecules like in your potatoes, in your pastas. Carbohydrates are composed of carbons, hydrogens, and oxygens, right, which is why it's a carbo, which is carbon, hydro, or hydra, which is hydrogen, and eights means oxygen. They have a general formula here of 1 to 2 to 1 ratio. So what that means is for every one carbon, you have double the amount of hydrogens and the same amount of oxygen. So here's a common example, a C6H12O6. Notice it's a 1 to 2 to 1 ratio. And this particular carbohydrate is glucose, right? If you haven't recognized it. Functions of your carbohydrates, they provide you that quick energy. So particularly if you're an athlete, right, and you're running, you stop and you take your little glucose pills and shots. Quick energy, it's the energy or it's the food source that your body uses first. It's real quick, it happens real fast. We can also use carbohydrates as raw materials, and we'll talk about those in a minute, and also for energy storage within our cells. The monomer of a carbohydrate is a saccharide, right? So a saccharide is a generic term for a monomer of sugar, for an individual sugar. And common example here is glucose. Most names for sugars end in an OSE, and generally they're classified by their number of carbons. So for example, this is a 6-carbon sugar. It's called a hexose, right? So hex means 6, it's an OSE. Common name is a glucose. 5-carbon sugar is a pentose, so pento means 5, such as fructose and ribose. 3-carbon sugar would be termed as a trio, so tri, like a tricycle, right, is 3 sugars. Example would be a glyceraldehyde, which we'll look at in a minute. Here's some common examples of the different types of sugars that we can have. So sugars can be classified as 3 different types. They can be monosaccharides, which are simple sugars, and mono means 1, so that means it's 1 sugar. A disaccharide, if there are 2 monomers, so that means there's 2 sugars that are linked together. Example is sucrose. Or anything over 2 is a polysaccharide. So just like a polymer, which is a generic term, a polysaccharide would be a large polymer of sugars. And common example here would be starch. So let's take a look at monosaccharides first. Monosaccharides are simple sugars that cannot be broken down by hydrolysis into smaller sugars. So they're as little as it gets, right? How do you get the rest of the energy out of this? Well, we're going to use a process called cellular respiration, which is in a future chapter. There's still lots of energy here, guys, between these bonds, and we will get it all, but just not right this moment. Hydrolysis doesn't get this energy. It only breaks the bonds between individual monomers. These are our main fuels for cellular work, as we said. And common examples, again, glucose is in your sports drinks. Fructose is natural sugar found in fruits, your apples, your bananas. Glucose and fructose are examples of something what we called an isomer. Isomers are molecules that have the same molecular formula but have different structures. So by molecular formula, it means they have the same number of atoms, they're just arranged differently. And we'll look at an example on the next slide. Some common monosaccharides that you should be familiar with as just examples of monosaccharides are, here's your fructose again, which we said is found in fruits. Here's a structure of a glucose and a structure of a galactose. You do not need to memorize the actual structure. I mean, you should be able to, for example, look at this and tell me, oh, that's a monosaccharide. You don't need to tell me which one it is. You know it's a monosaccharide because you only see one sugar here. But you should be able to at least list these common examples of monosaccharides. And there are some animations here more on isomers and additional information about carbohydrates. Here's a common example of those isomers that I just talked about. And so glucose and fructose. Again, if you look down here, here's their molecular structure. Both glucose and fructose have a molecular structure of C6H12O6. So they both have 6 carbons, 12 hydrogens, and 6 oxygens. The difference is where they're located. So in glucose, the carbon double bonded to oxygen is up here at the top of the molecule, whereas in fructose, it's actually a little bit farther down the molecule, which gives them different properties. So just by looking at this formula, I wouldn't be able to tell, right? I'd have to actually look at the structural formula in order to be able to tell the difference. Another common characteristic of monosaccharides is that generally in aqueous solutions, which an aqueous solution is just a water solution or a liquid solution, many monosaccharides form rings. So there's multiple ways that you can actually view and see this monosaccharide. So don't get confused if you see two different things. They're all monosaccharides. They just take on different structures and how they look. So this would be called the linear structure of glucose. Notice you still have your 1, 2, 3, 4, 5, 6 carbons. And you still have your 12 hydrogens and your 6 oxygens if you wanted to count. In an aqueous solution, these rings form spontaneously. So if you were to count, everything would be exactly the same. You still have your 6 carbons right here, right? But they've just formed a ring structure. Same exact thing. These 2 arrows right here mean that these molecules can go back and forth into different forms. And then this last one over here I wanted to show you is called the abbreviated ring structure. So you may start to notice that in future chapters. Anytime there's a hydrocarbon, a lot of times we don't even have to put the carbons right here. We assume that there's carbons, and technically I don't have to put off all of the hydrogens because I know that carbon can have 4 bonds, right? So I just automatically assume that there's carbons here, even though I may not list them all. All right, disaccharides. So we said, remember, that's a double sugar. There's 2 monomers. They're constructed from 2 monosaccharides. They're formed by a dehydration reaction. So we talked about that a few slides ago. They include lactose, which is found in milk, maltose, which is found in beer, malted milkshakes, candy bars, and sucrose, which is your common table sugar, right? You guys use sucrose all the time. Here's that dehydration we just talked about. Notice here's your OH group, here's your hydrogen group. What happens? You've released water, dehydration, synthesis reaction, or sometimes you may see the word synthesis added on there, sometimes not, and the bond is formed. So when I link a monomer of glucose with a monomer of galactose, I get a disaccharide of lactose, which is the sugar found in milk. The last type of carbohydrates that we mentioned were called polysaccharides. So poly, remember, means many. They're complex carbs. They're made of long chains of sugar units, polymers of monosaccharides. They cost very little energy to build. Some common examples that you should be familiar with is the first one on this slide is starch. This is used by plant cells to store energy, such as potatoes and grains. The second example listed here is glycogen, and this is used by animal cells to store energy, and it's easily converted to glucose when it's needed. And And the third example here is cellulose. So this is the most abundant organic compound on Earth. Forms these long cable-like structures within the walls of enclosed plants, and it cannot be broken apart by most animals. So, this is the fiber in your diet, right? So we lack a particular enzyme to break it down. So when you take fiber, right, it creates this bulk and helps you go to the restroom. And that's because we're not actually able to break this down for energy, right? So it stays in its initial form and moves through the digestive system as it is, or as its original form. Here's what they look like. You do not need to memorize the structures. Again, I just wanted to show you some examples. Your starch granules, so you can see these are long chains of glucose monomers. Your glycogen, again, which is found in animal cells for energy storage. You'll notice it's a little more complex. It branches off a little bit. And then eventually your cellulose molecules. So you can see it's actually parallel chains here, which are attached to each other through these cross links. So everything on here is a polysaccharide, and they're all linked together through dehydration, synthesis. They're all broken down through hydrolysis reactions. And remember that in each one of these bonds right here is where energy is stored, right? So if you don't use all of that glucose or all of that sucrose that you just ate in that candy bar, it's going to get stored in this form eventually, right? For short-term storage. Now, if you don't use it here, eventually the body's going to store it in the form of fat, right? Because that's for long-term energy storage. Here's some practice questions and some more. Go ahead and answer these. Make sure you have written down any questions that you may have as well. Now we're going to move on to our second macromolecule, which is the lipid. It says lipids are composed of carbons, hydrogens, and oxygens. They're not really macromolecules or polymers technically, but we're going to group them in for the purposes of this class. So we do build them. They're just not straight chains. They're all hydrophobic, which means they're unable to mix with water. And there are three different types right here. So we have fats, steroids, and phospholipids. And we're going to look at the function and the structure of each one of these. Here's an example of a common lipid. Notice oil, hydrophobic, hydrocarbons, and vinegar, which is hydrophilic, right? So that's why these do not mix. In this picture, non-polar at the top, polar at the bottom, right? Non-soluble at the top, soluble at the bottom. Long chains of hydrocarbons, these are probably ionic compounds that are able to dissolve in water. Let me clarify real fast again up here, so I know I may have some questions. It says not a macromolecule. Again, it depends on who you're talking to. We're going to classify it as a macromolecule. I usually take this sentence out. I forgot to take it out. Again, in the terms of this class, we're going to call macromolecules larger structures of smaller molecules, which is what we'll be building, as you can see in just a minute. So just eliminate that line right there. So fats, right? What's a typical fat? So remember there's three different types. The first type of lipid is a fat. A typical fat, or what we call a triglyceride, so tri probably means it has three of something, right? Consists of, here's your glycerol molecule right here, and 3 fatty acids. So this particular diagram only shows the first fatty acid chain. In the next diagram, we'll look at all 3 of them. But I want you to notice that right here, what's happening? Here's that hydrogen, here is that hydroxide or that OH, and what's being released? Water. Therefore, this must be a dehydration reaction, and a bond is going to form right there. Okay, and that'll happen here at this hydroxide and at this hydroxide. And again, the next slide we'll look at that more closely. The functions of fats, they are used for energy storage. So notice it says 2 times more than carbohydrates. Well, why? Well, because they have these long hydrocarbons. Review, hydrocarbons are covalent bonds. They're very stable. They don't mix in water. And guys, in between each one of these little chemical bonds are stored energy. So they're just really efficient at storing energy. That's why it's harder to get rid of fat, right? When you're trying to work out and exercise. They can also be used, or they are also used for cushioning organs within your body, and also for as an insulation factors, right? So think of like whale blubber, so you can keep the heat in and or keep the cold out. Are they polar or non-polar? Well, I just said, is this polar or non-polar? They are non-polar. Does that make them hydrophilic or hydrophobic? Makes them hydrophobic. They are afraid of the water. They do not mix with water. And here's an animation down here if you'd like some more information on that. This diagram again, here is my glycerol, my 3-carbon glycerol. Here's a single fatty acid chain. Here's an example of a dehydration reaction. And notice that down here in B, there are 1, 2, 3 fatty acids, which is why it's called a triglycerol or triglyceride, which is 3 fatty acids linked to an individual glycerol molecule. This is actually called an ester linkage, if you want to know specifically. And that's a linkage between this hydroxide and this COOH group right here. You don't need all that. It says a fat molecule, again, we generally will call, this is called the glycerol head, and you have 3 energy-rich hydrocarbon fatty acid tails. So again, tri, because there's 3, glycerol, because this is glycerol, right? And made through a dehydration reaction. If the carbon skeleton of the fatty acid has fewer than the maximum number of hydrogens, it is unsaturated. If it has the maximum number of hydrogens, it is saturated. So look down here, guys. If something is saturated, right, saturated, analogous would be like if you took a sponge and you put it into a bucket of water and picked it up and it was holding as much water as it could possibly hold, that is a saturated sponge. So a saturated fatty acid chain means that it's holding as many hydrogens as it can possibly hold, right? So there's no more room anywhere. An unsaturated means that, notice what happened here. You lost your hydrogen. Your hydrogen is now, well, the hydrogen's gone. That energy is now captured in the form of a double bond. So anywhere there's a double bond, this is an unsaturated because it's not saturated with hydrogens. This is a saturated. You'll notice when there's a double bond, what do you see? What happens to the shape of the molecule? It's bent, right? And so the fact that it's bent right here is not an accident, right? Form fits function. The form or the structural form that a molecule takes on will ultimately determine what its function is, generally. Hopefully you are familiar with saturated and unsaturated fatty acids. When you look on the sides of your food labels, underneath total fat, they will always list saturated and unsaturated, right? And so which one's better for you? Which one do you want to choose if you have to choose some type of fat? Notice there's 2 diagrams here. Here is my saturated fatty acids. Here are my unsaturated fatty acids. So here are my, here's my glycerol molecule. Notice that it's a nice straight chain. There's no kinks, right? When they are nice straight chains like this, they are all able to lay on top of each other very compact to form a dense solid. When there is a double bond, as we just saw, there's a kink. It causes that fatty acid chain to kink or to bend. Notice here's the bend. When it bends, they can no longer line up nice and tight. Therefore, there's that double bond. They can no longer form a solid, and they form a liquid instead. Looking at these 2, when you think about eating them, which one do you think is most likely to clog an artery? Well, that would be the saturated, right? So saturated fatty acids are known to contribute to something called atherosclerosis, which is a condition in which lipid-containing plaques can actually build up in the walls of your blood vessels. Your fish and your plant oils are your unsaturated, and I mean, it's common sense, right? I mean, this can obviously move much easier through your vessels, and it's not going to clog them or form plaques. So when you are looking at food substances, you're looking at your nutrition labels, generally you want to find the one that has the lowest amount of saturated fatty acids, right? It's these unsaturated, again, that form the kinks, and these are, again, your plant oils tend to be your unsaturated fatty acids. They tend to be the healthier, the olive oils and things like that. We talked about, I think on the first day, this process of hydrogenation. Remember when we talked about trans fats? It was one of our case studies in chapter 1. And remember when the nurses performed their scientific method, they had data that told them that there was a relationship between the number of heart attacks and the amount of trans fatty acids found in the cells or found in the lipid cells of the patients. And so they asked themselves the question, is there a relationship? The data supported it. And it ends up being a pretty well supported piece of data. So when you hydrogenate something, you convert unsaturated fats to saturated fats. So you actually make liquid fats solid at room temperature. This creates a trans fat, a type of unsaturated fat that is even less healthy than saturated fats. So again, if it's the cis process, if it's a natural process like this where the hydrogens are on one side, this is healthy. The body recognizes it as healthy. It's a healthy form of heart disease. When the hydrogens are on opposite sides and you force this hydrogenation, this is very bad. These types of trans fatty acids have been linked to things like heart disease, cancers, and it's because your body sees it as foreign. It's not a natural element. So your cells start to freak out and unusual things happen. Common examples, again, of your saturated fats over here. Common examples of your unsaturated fats over here. And you can see, here's what you look for, guys. Is it a natural unsaturated fatty acid, or is it artificial? And when you look, I know this is really small, but these are natural forms, right? Your shrimp, your omega-3s, your salmon, well, not the margarine, but in your plant oils. Fats, like for example, the shortening that was probably used to fry this donut. On the container, it usually says fully hydrogenated or partially hydrogenated. When you see that word hydrogenated, that means it has some unhealthy trans fats. You probably just want to stay away from those types of foods. We're going to move on to our second type of lipid now, which is called the steroid. Steroids are very different from fats in structure and function, but they're still classified as a lipid. Their defining characteristic is that they have this carbon skeleton with these 4 fused rings. So here they are, 1, 2, 3, 4. 4 fused rings. Steroids vary in the functional groups that are attached to these rings. So here's a CH3, here's an OH, here's a long hydrocarbon chain. So by changing what we call these functional groups that are attached, we actually change what the steroid is. 2 common examples of steroids, cholesterol and your sex hormones. Right? You guys are probably familiar with the artificial term of steroids where you're injected into your cells to grow big old muscles. But there are natural forms of steroids that are important for cells as well. Cholesterol, which is one of your steroids, is a key component of the cell membrane. So, you know, a lot of times you may have an elder or someone you know that has really high cholesterol and they may be on medication. But remember, there's good types of cholesterol and there's bad types of cholesterol. And you always need some type of cholesterol, right? And that's because, you can see as it says here, cholesterol is the base steroid from which your body produces other steroids, such as estrogen and testosterone, right? So estrogen is your female hormone, gives you your female characteristics. Testosterone is a male hormone, which gives males their male characteristics. And what I mean by that, if you look here, is the example. Here's cholesterol, so how you ate it, right, in your food in that big hamburger. And then through a series of chemical reactions over time, this 4-fused carbon ring obtains different functional groups. So if you're testosterone, here's what they look like. If you're estrogen, or this is a type of estrogen, Here's what they look like. So notice testosterone and estrogen look almost exactly the same. They vary here and they vary here. So it's just two little atoms that make them different, right? But they have huge differences on the effect of a body, right? Which are the main differences between males and females. Also, like for example, when we were talking about drugs and mimicking different types of hormones, progesterone is a type of synthetic hormone that is generally put in birth control pills. And for progesterone, you just change one or two of these functional groups, right, in order to make an artificial form. So the most important thing here is to know that, this is a steroid, and you can see this 4-fused carbon ring is the same. It's just these functional groups that change, right? And ultimately, it's classified as a type of lipid. Synthetic steroid, which is what you guys are probably familiar with, again, resembles testosterone, can mimic some of the effects of testosterone, but can also cause serious physical and mental problems. And as you guys probably already know, they're abused by athletes to enhance performance. And one of the common ones here is called THG, right? So these are people that have admitted to using it. But again, you can see here's that 4-fused carbon ring. All they had to do was change a little functional group in order to produce massive amounts of steroids, extremely similar to the one that your body uses, but not exactly. The last type of lipid that we're going to just brief on is called that phospholipid. So in the name tells you what it is, phospho, and it's a lipid. It is one glycerol molecule. So if you look in this diagram, here's that glycerol, that 3-carbon, the same one that was used in the triglyceride. But it has one phosphate. Here's a phosphate represented. You can see the P right there. And only 2 fatty acid chains. Remember in the triglyceride, it had 3 fatty acid chains. So some of this is quite similar. The difference is it has this nice little phosphate up here. Guys, this phosphate is negatively charged, as you can see. Therefore, it's polar. These long hydrocarbon chains are nonpolar. They haven't changed. So what ends up happening is you have this polar head area right here, and you have this non-polar chain area or tail area down here. And this molecule is what we call a phospholipid and has what we call a dual personality. So half of the molecule likes water. The other half of the molecule doesn't like water and doesn't mix with it, right? And this is very important, particularly when we talk about the cell membrane or the cell structure. Here's another picture of that phospholipid. It's just a different cartoon, polar head, nonpolar tails. And surrounding this cell, it's called the cell membrane. And when you actually zoom in on it, you can see there's actually a bilayer, right? So there's 2 layers here of those phospholipids. And the outside polar head faces the outside of the cell and the inside of the cell. And then there's this nonpolar hydrophobic middle region, which is very important. It protects the cell, right? It protects the inside of the cell from the outside. And in chapter 4, we'll talk very specific, or it might be chapter 5, we'll talk very specifically about this structure and how do we move things across and all that good stuff. For now, just know it's an example of a lipid. Some practice questions and a few more. Go ahead and pause, see if you can answer them. So far we've covered carbohydrates and the different types. We've covered lipids in the 3 different types. Now we're going to move on to our 3rd macromolecule, which are proteins. Proteins are polymers constructed of individual monomers of amino acids, right? So remember in your sugar, what was the monomer? It was a saccharide. In proteins, the monomer is an amino acid. They perform most of the tasks the body needs. They form enzymes, which are chemical changes that will increase the rate of reactions, and they're not changed themselves in the process. We have a whole chapter on enzymes later on. But down here you can see some major different types. Excuse me. They perform structural proteins like in your hair and the antlers. They can store different types of materials. They're contractile proteins, so like your actin and your myosin, responsible for muscle fibers. They can transport things throughout the body, such as oxygen. and they also act as enzymes. Again, enzymes are essential to life. Enzymes will speed up reactions, and without enzymes, your body would not be able to sustain life in general because the chemical reactions would move too slow. So some other examples, proteins, if you know anything about the immune system, they're your antibodies, and things like that. It's just a few examples. They're very important, guys. We used to think that DNA was the end-all, know-all, and after the Human Genome Project, now we know the whole sequence of the human genome, but we still have 5 million questions. And now we're looking for answers in proteins, and particularly protein folding. Turns out proteins are more important than the DNA code themselves. And we'll look at some examples of some of those mutations in future chapters. Today we'll just focus on the function and the structure. So as I said, the monomer of the protein is the amino acid. There are 20 different amino acids. And if you're a polymer, it's called a polypeptide. So remember in sugars, it was called a polysaccharide. For a protein, it's called a polypeptide. Proteins can be one or more polypeptide chains, folded and bonded. They're very large and complex, and this is the most important thing that you should remember about a protein. So they have a very large, complex 3D shape. Proteins are only functional if their shape is proper. Shape is very important in proteins. And we'll look at some examples in future slides. Here's an example of 2 amino acids. What do you notice? Here's the OH group, here's the hydrogen group, and what have we released? Water. So what type of reaction is this? Dehydration, and the bond is formed. Same reaction used to build triglycerides, same reaction used to build polysaccharides. The structure of the individual amino acid is unique. So you can see each amino acid consists of a central carbon right in the middle, bonded to 4 different partners. So every single amino acid has a central carbon, has an amino group, which is an NH2 group, has a carboxyl group, which is a carbon double bonded to oxygen, and an OH group, defined as a carboxyl, has a hydrogen, and they all have this side group over here. So side groups, also known as, you may see it written as an R group or a side chain or a variable group. These confer unique chemical properties to that amino acid. So again, this portion right here, exactly the same in all amino acids. The only thing that differs in an amino acid is this. This is what makes them all special. So what type of properties do those side chains have? Well, let's look at these 2 examples. Notice both of these amino acids have central carbons. They both have amino groups on the left side. They both have carboxylic acid groups on the right side, but they have different R groups or side chains. Leucine and serine, these are just 2 different examples of amino acids. This is a long chain of hydrocarbons. So that means what? He's hydrophobic. Does he like the water? No. This is a hydrophilic side group, and it's hydrophilic because it has an oxygen right here. Does he like the water? Yes, he does. So what happens? This amino acid is going to want to stay away from water. This amino acid is going to want to interact or face water. These hydrophobic and hydrophilic differences between the amino acids are going to cause the protein to fold in a specific way, right? Remember, 72% of the cell is made up of water. So where is this portion of the protein going to go? Well, he's probably going to try to fold on the inside. This portion of the protein is probably going to try to form the outside, right? Here's some other examples. I just wanted to show you. You do not need to memorize these, but I just wanted to show you some other nonpolar examples. And also just to kind of give you a list of the different types of amino acids there are. Guys, also generally, we abbreviate amino acids with these 3-letter abbreviations. So glycine's abbreviated GLY, and so on. Alanine, valine, leucine. And you can see the highlighted side chains. These are all nonpolar because they're all hydrocarbons, so they're all afraid of the water. Here's some examples of polar, charged, or hydrophilic. You can see the oxygens, oxygen, sulfur, and so on. Anything that has a positive or negative charge, they're all going to like water, right? So all of these amino acids are going to try to interact with water when they're folding together. Proteins form polymers, just as your saccharides do, right? Except they form peptide bonds in polypeptides instead of glycosidic bonds for polysaccharides. Here is the general reaction that we just saw. Amino acids. Here's our hydroxide, our hydrogen. We release water, this dehydration reaction. And here's the peptide bond. Again, this is called a peptide bond because it's between two amino acids. If it was between two saccharides, it would be called a glycosidic bond, right, for glycogen. So your body has tens of thousands of different kinds of proteins, and their arrangement of amino acids, as well as how they fold upon each other, is very important to the structure and the function of that particular protein. So this structure is called the primary structure. It just basically looks like a bunch of beads, right? And this long string of amino acids. Here's a very common example, guys. A slight change, a single nucleotide change in a DNA sequence causes a single amino acid change. So we go from glycine to valine, one single amino acid chain in that whole protein. And it causes the red blood cell to go from this nice donut shape to this sickle cell. And so the hemoglobin has this nice area in the middle where it actually carries oxygen to other parts of all the other parts of your body. The sickle does not have that donut shape, right? So the actual form is altered because it's mutated. So he can no longer bring oxygen to other parts of the body. Again, a single amino acid. Now what's the difference between glycine and valine? The side chains are different. This one's hydrophilic, this one's hydrophobic. So a single change in a side chain caused these two proteins to misfold. So they fold it completely different. And now you have detrimental effects, right? You're not able to deliver oxygen to other parts of your body efficiently. To reiterate, protein shape. So a function of a protein consists of one or more polypeptides. Again, guys, it could be multiple polypeptides that are linked together. So your examples down here, these nice crystallized graphics, these are actually multiple polypeptides that have all come together. One polypeptide has 3 levels of structure, and then you can have more than one polypeptide, which can eventually move into a quaternary structure. Structure equals function, and the 3D shape is essential. There's quite a few videos here that you can watch and go back. I will go through each one of these structures that I'm about to talk about. Here's your primary structure. So we're going to go over again the different structures, all the different levels. The primary structure of a protein is simply the chain of amino acids. It's not folded. It's completely non-functional. It can do nothing within the body because it is not folded. After the primary structure has been formed, these amino acids will then interact with each other to form a secondary structure. There are 2 secondary structures. The first one is called the beta-pleated sheet, and the second structure is called the alpha helix. So again, it's the interactions between the amino acids and the side chains that cause these structures to occur. After the secondary structures have been formed, next the protein will form what we call the tertiary structure, in which the remaining folding of the protein will occur. Notice that here's your beta-pleated sheets, looks like a pleated sheet. Here's your alpha helix, looks like a helix, a cylindrical structure, are still maintained within the tertiary structure. So it's basically the secondary structures folding upon each other. At this level, the tertiary level, the protein is fully functional. Some proteins may need to form or may need to coincide and work with other proteins. This quaternary structure is not necessary in all proteins. It is necessary in the example that I showed you earlier in hemoglobin. So hemoglobin is actually made up of 4 different proteins or 4 different polypeptides that work together. But again, non-functional, non-functional, This is the first time you have a functional protein unless he needs to work with other proteins, which that would be your quaternary structure. But proteins don't always have to take on this quaternary structure. A protein's three-dimensional shape will recognize and bind to other molecules, which ultimately enables the protein to carry out a specific function, which I just said. You can see here's an actual protein. This is maybe something that the protein is trying to interact with. And if you were to actually zoom in on here, you would see that this particular substrate will fit exactly like a lock and key method into this protein. So if there's any alteration of the shape of this protein, is he going to fit in there? No, right? And then the reaction can't occur. So shape is extremely important. What determines the protein shape? So protein shape is very sensitive to the surrounding environment. Guys, remember when proteins are in a cell, there's water and ions and all kind of other crazy things happening in the cell. So unfavorable conditions such as high temperatures, pH changes can do something called denature the protein. Here is a picture of a denatured protein. So here's the folded protein. When you add high temperatures or you change the pH or you change the salt concentration, you denature that protein. Is this protein functional anymore? Nope. It goes back to its primary structure, that chain of amino acids. He no longer works, right? High fevers above 104 can sometimes cause proteins to denature, so can high salt concentration, any changes in pH. Remember in pH, the hydrogen ion concentration. Again, you're interacting here. Anything that's going to interact with all of these amino acid side chains can ultimately change the shape. Anything that disrupts bonds. Some examples of misfolded proteins that are associated with some diseases like Alzheimer's disease, mad cow's disease, and Parkinson's disease. So sometimes they can go back to a nature state. Sometimes they're permanently left this way. So your common example would be a raw egg, right? The white clear part on the outside of the yolk is a protein. That's where all the protein is. That's why eggs are packed with protein. When you cook it or you fry the egg, what happens? It becomes like that white hard color. It becomes that color because you have denatured the protein. Can you go backwards? No, you can't go back to the original egg, right? And how did you denature it? Well, you added it to a hot skillet, right? You've added a high temperature, so you've denatured that protein. It's no longer functional. Here's some practice questions. Pause, answer them. All right, here's our last macromolecule that we're going to talk about today, and this is the nucleic acid. Function of the nucleic acid is to store and transmit genetic information. Where's it found? Remember when we talked about in chapter 1 that genetic information is stored in the nucleus, right? The DNA, only the DNA actually. Well, no, RNA too, never mind. There are only two examples of nucleic acids. The first one is RNA, which stands for ribonucleic acid. The second example is DNA, which is that genetic information stored in the nucleus of your cells. DNA stands for deoxyribonucleic acid, and the structure or the monomer is the nucleotide. So the sugar monomer is a saccharide, the protein monomer is an amino acid, and the nucleic acid monomer is a nucleotide. And remember, when you link those monomers together through dehydration synthesis reactions, you get polymers, right, or larger macromolecules. Here's some examples. Here's your RNA molecule. Here's your DNA molecule. You'll notice right away there's a few differences. DNA is double-stranded helix. RNA is only single-stranded. They both have a sugar phosphate. They both have base pairs. They both have nitrogenous bases, and by definition, nitrogenous base are one of these. You'll notice there's a few differences in these, though, as well. They both have cytosine. They both carry guanine. They both carry adenine. But RNA has a uracil in it, and DNA has a thymine in it, right? And if you actually look at the structure, you'll notice there's different side chains over here coming off. DNA resides in cells in long fibers called chromosomes. Again, remember, location is within the nucleus of the eukaryotic cell. A gene, as you see right here, right, it's a short section of DNA. specific stretch that programs amino acids to eventually form protein. So here is our macromolecule that we were just talking about. The central dogma right here is going from DNA, translating that code into RNA, and then eventually translating that code into protein. And this process right here we will talk about very specifically in future chapters. But both of these are called nucleic acid. Both DNA and RNA are called nucleic acids. Nucleic acids are polymers of nucleotides, and each nucleotide has 3 different parts. So as you look at your nucleotide down here, each nucleotide has a 5-carbon sugar. So here's your sugar, 1, 2, 3, 4, 5. It's a ribose sugar in RNA. It's a deoxyribose sugar in DNA, and we'll talk about that structure more specifically in future chapters as well. Each nucleotide is also made up of a phosphate group over here, which is represented as the P, surrounded by the 4 oxygens and the negative charge. And then they each have a nitrogenous base. So your nitrogenous base for DNA can be that A, G, C, or T. So that adenine, guanine, cytosine, or thymine. Symbols used in your book may look like this, right? So it's not as molecular: a sugar, a phosphate, and a base, right? Or nitrogenous base. More specifically, you can actually see the structures here. Each DNA nucleotide has one of the following. So you'll notice adenine and guanine are actually double ring structures. So notice there's 2 rings and 2 rings here. And thymine and cytosine are actually single ring structures. So there are some differences in these nucleotides. Notice down here in this double helix, the blue-yellow is actually the repeating sugar phosphates, and in the middle is where those nucleotides are located. Dehydration reactions, as we said, link these individual nucleotides together. So by releasing water, they form covalent bonds. And as you begin to link these nucleotides together, you get this sugar phosphate backbone. So here it is: sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, right? And then sticking out from that sugar phosphate are the bases, right? So the A, T, G, and C's, or the uracil. When you put 2 strands of these DNA molecules together, you get what we call a double helix, and this is the natural form of DNA, which is found in your nucleus. You'll notice that here is your sugar phosphate, so there's 2 strands of sugar phosphates. In the middle of the double helix, there's your nitrogenous bases, and you should notice that A always binds with T, and G always binds with C. Always, always. So in this way, we're able to make predictions about DNA sequences, right? If I have a C here, I know on the other side there's a G, and vice versa. Why do they only pair? Why does A only bind with T and G only bind with C? Well, again, it's based on structure, right? So A only binds with T and G only binds with C. A double ring structure only binds with a single, and a double binds with a single. It has to do with spacing. If these 2 try to bind together, the form of the double helix would be wrong. It wouldn't be the correct structure. So due to the lining up of the nitrogenous bases, as well as these hydrogen bonds that form right here, they may only bind with specific nucleotides. Here is a molecular, you can see, of your sugar phosphate backbone: sugar, phosphate, sugar, phosphate, sugar, phosphate. Remember, these were nucleotides. Here's a nucleotide. They were linked together by dehydration synthesis. A binds with T, so adenine binds with thymine through 2 hydrogen bonds. Remember, we talked about hydrogen bonds. What are they? There's a slight positive, slight negative charge that's attracting these 2 together. Guanine binds with cytosine and forms 3 hydrogen bonds right here. Again, if you were to try to put these 2 together, they would not line up properly, right? So they cannot bind with each other. Woo. What happened there? Copying of our DNA molecule. The two strands of DNA are complementary to each other. So by complementary, it means that you can make predictions of what the other strand is. Complementary means that A always binds with T and G always binds with C. So they complement each other. If you have one strand of DNA, if I know the sequence of one strand, I can predict the sequence of the other one based on the base pairing rules. This is a great system because it helps to ensure that the DNA will be copied exactly the same way as it moves on to offspring, right? So it's kind of like you're double coding that particular genetic information. When in life of a cell does replication occur? It occurs in mitosis and meiosis. And that's in future chapters. We'll talk about how we replicate this DNA and how do we pass this DNA onto our offspring into other cells, right, in cellular division and replication. Replication says it has not escaped our notice that specific pairings we have postulated immediately suggest a possible copying mechanism for the genetic material. This is the famous Watson and Crick. These are the guys who first published the structure of DNA as a double helix. And this is telling us that as long as you have an old strand, you can make a prediction of what the new strand is going to be, right? So if I know the old strand has a C, I know the new strand is going to have a G. The old strand has an A on it, I know the new strand is going to have a T. We spent a lot of time talking about DNA. We're just going to touch on real quickly, as we did a few slides ago, just a few differences between DNA and RNA. So first of all, the name, right? RNA is ribonucleic acid as opposed to deoxyribonucleic acid. RNA is usually single-stranded, DNA is double-stranded, and RNA uses the sugar ribose and the nitrogenous base uracil instead of thymine, right? And so your ribose sugar, it's a 5-carbon sugar, 1, 2, 3, 4, 5, but it has an OH group right here. Does everybody see that? Here's your OH, ribose. Deoxyribose, let me show you that one. Oh, never mind, I can see it right here in the next slide. I'll show you in the next slide. So ribose is this normal 5-carbon sugar with a hydroxide. Deoxy, what do you think that means? If you de something, you take it off. So a deoxyribose for DNA, it's probably going to be missing the oxygen right there, right? We'll look at the structure in just a minute. Just to further clarify your types of nucleotides, or your nucleotides which can make up, or your nitrogenous bases which make up your nucleotides. There are purines and pyrimidines. You don't need to memorize them. I just want you to know that there's a difference. Your double, I'm paying attention to something else. Your double ring structures of adenine and guanine, and your single ring structures, which are called pyrimidines of cytosine, thymine, and uracil. This is a nice diagram that kind of shows you the big overall picture of the difference between DNA and RNA. So here are the individual nucleotides. What do you notice? Here's your sugar. They both have 5 carbons, so 1, 2, 3, 4, 5, but what's the difference? The sugar here is ribose. Ribose has an OH. Deoxy, notice they're missing an OH right here. So everybody see that? Deoxyribose. They both have phosphates, and they both have nitrogen bases. However, for DNA, it has a thymine base as a pyrimidine. For RNA, it has a uracil base as a pyrimidine. So in DNA, A binds with T, and G always binds with C. But in RNA, A will actually decode as uracil, and G will decode or bind with cytosine. Oops, not done there. DNA is also a double helix, as you can see. There's actually 2 strands here. The outer sugar phosphate backbone and the inner portion of that DNA is made up of those A, T, G, and C's held together by hydrogen bonds, whereas your RNA is single-stranded. It has a single sugar phosphate bond. Does not have hydrogen bonds in the middle because it's only a single strand, and it has this uracil right here. Lots of practice questions coming. Practice. Nice little review of what we just talked about. Here's our dehydration reactions to build polymers, releasing water. Our hydrolysis reaction uses water in order to break the bond apart. In a dehydration reaction, water is released in order to build that polymer. Here's a nice review of all of the different macromolecules that we talked about today. So your carbs, your lipids, your proteins, your nucleic acids. I'm not going to go over this information right now, but you should definitely know the function of each, major components, and common examples that are talked about here, as well as the ones that are talked about in the PowerPoint itself. Carbohydrates, your lipids, long-term energy storage, your glycerol and fatty acids. Now there's an example. Here's your fats, steroids, and you should also add phospholipids to this example. I've added that one to this particular chapter. Your proteins, remember multifunctional, 3D shape is very important. Your basic monomer is your amino acid. And last, your nucleic acid, so your DNA and RNAs, those individual monomers of a nucleotide, which is made up of 3 parts, your sugar, your phosphate, and your base. Very important in this chapter is also protein folding rules. And so to go from your primary to your secondary structure of alpha helices and beta pleated sheets, into your tertiary structure, which is your overall functional structure, meaning that the protein can be fully functional unless it needs the help of other proteins, and it must take on a quaternary structure. We briefly also went over the differences between the DNA, nucleotides, a DNA strand, what makes up a DNA strand, and then we also talked about today 3 specific parts of a DNA nucleotide. There is a lot of practice questions. Take your time, go through them, answer them all. A few more. Remember guys, practice questions, you will see them on your test. You will see them maybe not exactly as they're written here, but they'll be very similar. And then here's a challenge question. And take a read. Remember, I will definitely ask you the challenge question in class. It's always worth more than 1 optional point. So take a minute to really answer this question. So that way, if you're not sure and you can't answer these questions right now, then make a note of it. Put a little star next to it. So when I brief you and go over the talking points of chapter 3, you have a little star and you know to ask me during the lecture time. Goodbye.